Hi, this is Ms. Delosier, and this is our continuation of our long metabolism review. We are on questions 11 through 20-ish on this section. Number 11, where are the proteins in the electron transport chain located? So let's do a real quick review. This is our kind of jacked up looking mitochondria. And mitochondria have a they have two membranes. They're dual membranes, so they have an outer membrane that's on the outside, inner membrane that's on the inside. They've got all the surface area to go, all the folds are there to increase surface area so you can do more electron transport. The electron transport chain is located here on this membrane, okay? And that is the inner membrane. This is the matrix, and this is the intermembrane space, okay? So outer membrane right here, inner membrane right there. Matrix is the gooey inner center, and the inner membrane space is just the space between the two membranes. So where are the proteins of the ETC located? And that's going to be on the mitochondrial inner membrane. So that is C. Number 12. In cellular respiration, the energy for most ATP synthesis is supplied by, so where am I getting most of my ATPs? Most of them are going to be made electron to the next one, so that's going to be during the electron transport chain. So something to do with the electron transport chain. High energy phosphate bonds in organic molecules. I'm making high energy phosphate bonds. I'm adding that other one on, but that doesn't sound right. A proton gradient across a membrane. Yes. Converting oxygen to ATP. No, that's impossible. Transferring electrons from organic molecules to pyruvate. No, really, we're using the pyruvate to transfer the electrons to the high energy electron carriers, NADH and FADH2. Generating carbon dioxide and oxygen in the electron transport chain. I'm not generating oxygen in the electron transport chain. I'm going ahead and I'm combining oxygen with electrons, and I'm actually making water here, and then the carbon dioxide is actually made during Krebs. So my answer here is B. And again, this is, for those of you that didn't watch the first video, a proton gradient across a membrane then leading to ATP synthesis is called oxidative phosphorylation or chemiosmosis. So both of those are the same thing as this proton gradient across a membrane. So let's go ahead and clear my ink layer and then scroll down, and let's work on 13 through 16. The primary role of oxygen in cellular respiration is to yield energy in the form of ATP as it's passed down the respiratory chain. So I'm not passing the oxygen, I'm passing the electrons. So that sounds kind of smart, but it's a little off. Is it to act as an acceptor for electrons and hydrogens? Yes, oxygen is demanding electrons. That's the whole thing it wants to do. So that sounds true. I might not remember the part about water, but that sounds true. Combined with carbon, forming carbon dioxide. No, that's not the primary role. Combined with lactate, forming pyruvate. That is not my goal. My goal is to not involve lactate at all in cellular respiration, because lactate is fermentation. Catalyze the reactions of glycolysis. Glycolysis is anaerobic. It requires no oxygen. So that is false. So my answer here is going to be B. Okay. 14. Energy released by the electron transport chain is used to pump hydrogen ions into which location in the eukaryotic cells? So remember, I've got my electron transport chain here, okay? So my electron transport chain is here. So I'm actually pumping the hydrogens into this space right here, into the space between the membranes, in the intermembrane space. And then what's going to happen is they're going to flow through the ATP synthase, and that's going to produce the ATP in the matrix. But my answer is the electron chain is used to pump the hydrogens into the intermembrane space, C. So where's ATP synthase located in the mitochondria? It's located on the inner membrane. And then approximately how many molecules of ATP are produced from the complete oxidation of 2 glucose molecules? So 1 glucose gives you approximately 36 ATP. It's really anywhere from 34 to 38, and it really depends on a lot of different variables that I don't even want to talk to you about because they'll just kind of confuse the matter. But what that means is that my answer for 2 glucoses is going to be 2 times approximately 36, and that's going to give you somewhere in this upper range. I mean, it's not going to be exactly this, and the deal is that you're actually using part of those for other cellular processes. Like we talked earlier about how do you get the pyruvate in to the mitochondria, and the pyruvate has to be moved into the mitochondria through active transport. Well, active transport requires ATP. So when you look at the net cellular product, you end up using, that's kind of why it's waffly. And I didn't want to talk about that, and I'm just going to shut up about it now. Number 17 and 18. Which of the following normally occurs regardless of whether or not oxygen is present? Glycolysis occurs when oxygen is not present. Fermentation is a no oxygen. Oxidation cannot occur when there's no oxygen present. Okay, it can't. And yeah, it can't. So it's A. And then the citric acid cycle actually does use oxygen also, because you combine with the you combine the carbon with oxygen to make CO2. It's not its primary role, but it does happen. Number 18, which of the following occurs in the cytosol of a eukaryotic cell? Glycolysis, yes. Fermentation, also yes. Fermentation, yes. Chemiosmosis, nope, that is the mitochondria. Oxidation of pyruvate to acetyl-CoA, that is in the mitochondria. Citric acid cycle, that's Krebs, you guys. We already said that. So that's the mitochondria. And then oxidative phosphorylation, that's electron transport. Just for the record, Krebs happens in the matrix. I don't know if we talked about that or not. I don't think we did. So my answer here is A. And moving on to the next one. Number 19 and number 20. In the absence of oxygen, yeast cells can obtain energy by fermentation, resulting in the production of fermentation. So it's got to either be lactic acid or ethanol. So it's not pyruvate, it's not oxygen, and I told you didn't have to know acetyl-CoA. So it's not that. So it's either lactic acid fermentation or ethanol fermentation. And if you think about it, yeast are involved in the production of beer, so this has to be ethyl alcohol fermentation. 20. Why is glycolysis considered to be one of the first metabolic pathways to have evolved? Because it produces much less ATP than does oxidative phosphorylation. That's true, but that is not a reason why we think it evolved first. It does not involve organelles or specialized structures. True. Does not require oxygen. Also true. And is present in most organisms. Also true. It is found in prokaryotic cells, but not eukaryotic cells. That is false. It relies on chemiosmosis. That's false, because chemiosmosis is not involved in glycolysis. It requires the presence of membrane-enclosed organelles. False. So glycolysis, we think, is the oldest one because everything has it. It doesn't require any specialized structures. And if you guys think about it, plants had to exist as some type of organism before the chloroplast was brought in. So photosynthesis happened after cells happened, so you can't have oxygen if there's no photosynthesis. So anyway, it's B. And we're going to actually do 21, and then I'll move on from the next ones in the next video. When electrons flow along the electron transport chains of mitochondria, which of the following changes occur? So when the electrons flow across the electron transport chain of mitochondria. So this is the electron to the next one. So remember, you've got your stable protein, and it's going to pass it off, and then another stable protein, and then it's going to pass it off. So this is that long chain that we talked about. Ignore this. That long chain that we talked about. And we're going to go ahead and we're going to look at that. So as I do that whole thing, I'm going to pump hydrogens in, and I'm going to pass the electrons down, and then eventually I'm going to make an ATP, right? So if I pump the hydrogens there, I'm going to go ahead and I'm going to be increasing the pH in the inner membrane space. ATP synthase pumps protons by active transport. No, because the hydrogens are going to flow through facilitated fusion. The electrons gain free energy. The electrons are actually losing energy. NAD plus is oxidized. That would mean that I'm forming NADH, and I'm actually going from NADH to NAD plus here. And so I'm not oxidizing it. The cytochromes phosphorylate ADP to ATP. That's not true. I make the ATP using ATP synthase, which only leaves me the pH of the matrix increases, which is actually false. This is actually a typo. This is supposed to say oh no wait, that's true. Sorry. I was thinking the acidity. So the pH of the matrix increases. I'm going to add a bunch of hydrogens here. So this is going to become more acidic. This is going to become more basic. And basic would be like 14 and acidic would be like 2. So yeah, the pH of the matrix increases. Sorry, I got distracted by being rushed for the bell. Anyway, I will continue with number 22 in the next video.